The myofibrils are organelles that are composed of repeating units that are chained end to end, and those repeating units are called sarcomeres. The sarcomere is the functional unit of skeletal muscle contraction. Sarcomeres possess two major types of protein filaments. Those two types are the thick myofilaments and the thin myofilaments. The thick myofilaments are constructed from a protein called myosin. Myosin is a club-shaped protein with the myosin heads containing binding sites for both actin and ATP. The tails of the myosin proteins are twisted together, and then many of those myosin proteins are bundled together to form the thick myofilament. The thin myofilaments, on the other hand, are made of actin proteins with two regulatory proteins that are associated with them. The regulatory proteins are called troponin and tropomyosin. The actin proteins are bead-like, and they possess myosin binding sites. The thin myofilament is composed of two strands of actin proteins that are twisted together. The troponin anchors the tropomyosin, and the tropomyosin is what covers the myosin binding sites that are found on the actin proteins. Figure 10.5 examines the major components of the sarcomere. On the left, we see the thick filaments, which are composed primarily of myosin molecules. The myosin heads of the myosin proteins possess binding sites for actin in addition to possessing binding sites for ATP. On the right, the thin filaments are shown, which are composed of actin, troponin, and tropomyosin. The actin proteins are bead-like with two strings of actin proteins twisted together. The actin proteins possess binding sites for myosin. Covering the myosin binding sites is tropomyosin, which is a filament-like protein, and then we have troponin, which supports the tropomyosin. The Z-discs, which are also called the Z-lines, form the ends of the sarcomere. They also function to anchor the thin myofilaments, with the thin myofilaments projecting towards the middle of the sarcomere. The A-bands of the sarcomere represent regions where the thick myofilaments and the thin myofilaments overlap, and due to this overlap, the A-band appears dark under the microscope. The I-bands of the sarcomere represent regions where the Z-discs and the thin myofilaments are present, but they have no overlap with the thick myofilaments. And because of this lack of overlap, the I-band region appears more lightly colored under the microscope. Returning to figure 10.5, the Z-lines or the Z-discs form the ends of the sarcomere with additional sarcomeres present on either side of the focal sarcomere in this diagram. The I-bands represent the place in the sarcomere where only the Z-discs and the thin filaments are found. Meanwhile, the A-bands are found in the center of the sarcomere, as it represents the location where the thin filaments and the thick filaments overlap with one another.